You're tuned into a discussion of anatomy and physiology, and we've proceeded as far as Chapter 4, which is a survey of all the tissues of your body that make up all the organs and organ systems. So we're going to work our way through all the different tissue types and find out how they're constructed and what their properties are, so that when we go to talking about anatomy of larger systems and organs, we'll understand those properties inherently. So that's what we're going to try to accomplish, learning a little bit about each one of those tissues so that we understand not only the anatomy of the systems and the organs as we learn them, but what they do. The physiology is based on what the properties of the structures are. So we learned that there are many different types of cells in your body, each of which is specialized for a particular function. And when you take a group of like cells, that's what we define as a tissue. So we'll be looking essentially at what some different cell types are like, and then what tissues they make in composite. We'll also be looking at some tissues that really aren't very cellular at all. They're constructed by certain resident cells, and quite a lot of building material is actually present. We'll be using histology to identify the appearance of various tissues, and so we'll be looking at thin sections that have been cut from various tissues and then fixed, stained, and then made images, micrographs of. So the four types of tissues that make up all of the organs of your body are listed here: nervous tissue, muscle tissue, epithelial tissue, and connective tissue. Nervous tissue produces a organ system that is a controlling system for many parts of your body. The nervous system includes the brain and spinal cords and the peripheral nerves. Muscle tissue allows for movement of blood through the circulation, movement of the skeleton, movement of materials within hollow organs of our body. Epithelial tissues form boundaries that separate different departments, largely separating our body from the outside world, and we'll see some of the examples of that. Connective tissues are interesting. They are the structural components of our body, the skeleton, the bones, all the tendons and ligaments that connect the bones together and connect the muscles to bones. The blood itself is a type of connective tissue. So those are the four basic tissue types that we're going to be looking at as we proceed through this chapter. We'll start with epithelial tissues. So epithelial tissues are sheets of cells stitched together with cell-cell junctions that form boundaries between our bodies and the outside world, or between different body cavities. So we have covering and lining epithelia, which is largely what we'll be talking about, which form these boundaries. Or there's also some modified epithelial cell types that produce secretions. Exocrine glands specifically produce secretions, and they are made up of epithelial cells. So let's take a look at some specific features of epithelia. Come back to that last section of the previous slide. Epithelial cells, and therefore the epithelia themselves, the sheets of cells, are polarized. That means that the cells are asymmetrical. They have different receptors on the basal surface, where they attach to the body tissues, and different receptors on the apical surface, typically where they're free and exposed to some hollow organ in the body or some exterior surface of the body. Epithelial cells have special cell-cell junctions or contacts that hold them together and hold them to the tissues that they adhere to. And they also usually have some tight junctions that control the movement of substances across the epithelial boundary. And they're attached. What are they actually attached to? Connective tissues. So we'll have to wait for a little while to see what it is that underlies an epithelial layer, but it turned out to be very important for the survival of the epithelium, but it's going to be a type of connective tissue. And a final thing we should quickly note about epithelia before we go on is that they have no blood vessels of their own. They may have some sensory neurons to allow us to gain some information about stimuli or about signals that are or things that are impacting them, but they have no blood vessels, hence all their nutrients and oxygen come from the connective tissues in the vicinity. Epithelial cells are inherently well regenerated, meaning the cells can replace when there's a loss of tissue. If there's a tissue injury in which now there's an absence of epithelial tissue in some location, It's a very routine process for some new cells to be produced by mitosis and to re-epithelialize that region. There is a limit, but as we'll see, especially when we talk about the skin, we encounter this limit occasionally, but there's an amazing regenerative capacity. The origin of all those cells of an epithelium are stem cells that lie in and among the rank and file cells of the epithelium. Stem cells are some very specialized cells that have some unique properties that allow them to fill this role. One is that they're immortal. Most of the cells in our bodies can only divide so many times before they age out, essentially become senescent. That means they can, say, divide maybe 50 times, and they essentially have a built-in timer, and after that they can't divide anymore. But stem cells can divide indefinitely, so they can continue throughout your life to act as a source for all those new epithelial cells to replace those that are lost, or some epithelia just continue to rebuild themselves and replace themselves. Stem cells divide only occasionally, and they divide by asymmetrical division, as we'll see, but the daughter cells that actually give rise to all the epithelial cells that we're going to need divide very quickly and can provide a lot of cells to fill in a void or to replace a normally replaced epithelium, regenerated epithelium, but the stem cells themselves only divide occasionally. When they divide, usually when you've studied mitosis, you'll see that the cell enlarges, it grows, and then it doubles its DNA complement. It copies all the chromosomes, separates them into two identical groups of 46 chromosomes, and then eventually the two new daughter cells separate and you wind up with two identical cells. And that is typically the case, but when it comes to stem cells, the stem cell must regenerate itself. It must recapitulate another stem cell, and the daughter cell is different from the stem cell, so we have what's called asymmetric division. Stem cell must always be retained, or we won't have an immortal cell at all. So let's talk about the actual epithelia. And we'll go through a process, a survey of all the epithelia, and we'll try to do it in an orderly way. We'll observe that epithelia are classified according to two criteria: the number of layers and the shape of the cells that make up the epithelial tissue. So when it comes to the number of layers, our epithelium can be either simple if it has one single layer, or if it has more than one, no matter how many layers. It's called a stratified epithelium. The cells that make up the epithelium contribute to the naming and classification. The ultra-thin cells, thin flat cells, are called squames or squamous cells. Chunkier cells that essentially are like a cube are called cuboidal cells, and tall rectangular epithelial cells are called columnar cells. So let's try and see how many combinations of those two different criteria we can find in the body. We're going to try to be able to find examples of as many as possible that have physiological relevance. As you'll see, we're going to skip over a couple of categories in which there are epithelia to be found, but not really any known physiological advantage. All right, let's start out with a simple squamous epithelium. A simple squamous epithelium is ultra-thin. One of the skills you need to acquire in learning to be a good histologist is looking at something in 2 dimensions and then imagining what must have been present in 3 dimensions to give rise to that appearance when cut through with the histologist's knife. So here we have the edges or the walls of air sacs in the lungs, microscopic air sacs called alveoli in the lungs, and they are essentially spherical, but when we cut through them, we wind up with circles. So that's the kind of connection you have to make. They're a little shriveled up because of the processes of fixing and staining the tissue, produce some changes that we call artifacts. Anytime there's a change in the tissue from the way it looked in life to the way it looks in the fixed and stained section, we call it an artifact. Anyway, these are the air sacs, and they're ultra-thin walled. And you can actually look in here, if you look carefully, you can see some bright red spots. Those are red blood cells. They're different in color from the stain that was used to make these cells show up on this slide. And so all these represent blood capillaries. Think of how amazingly small and thin-walled they are. And so the distance over which oxygen must diffuse to go from the air inside the alveolus into a red blood cell in this capillary is unbelievably small, and therefore the saturation of a red blood cell with oxygen can occur in maybe as few as 20 milliseconds as the blood cells course through a capillary in the wall of an alveolus. So these cells are very, very thin, specifically to allow for diffusion to happen efficiently. So they're specialized for diffusion. In addition, I will just mention, before we go on, the capillaries themselves are single-cell thick tubes, and these cells are also simple squamous epithelial cells, and they have their own name. The cardiovascular system is a very sort of heralded system in its importance in healthcare and in the history of anatomy and physiology, so everything in there gets its own names. And so the epithelium of the capillaries, and indeed the lining of all the blood vessels, which is the same type of epithelium, is called the endothelium. We'll probably come to that in just a little bit, but if I don't remember to mention it later, it's the endothelium that makes up the capillaries and tiny venules, and then that same type of cell lines all the larger blood vessels. Ultra thin for diffusion. How about a chunkier cell? These cells are simple cuboidal cells of the kidney tubules, and the way the kidneys work is a bit unusual, but it involves having to transport a very large amount of solutes and water that's already been essentially released from the blood into a container, into the capsule of the glomerulus, and then we're going to reabsorb 99% of all that water and solutes, one molecule at a time with the solutes, and water will follow by osmosis. That's a big job of transport or absorption, and so these cells are specialized for active transport. This set of simple cuboidal epithelial cells needs some more volume to be able to contain all the mitochondria it takes to make enough ATP to drive active transport, we call it. We're going to invest ATP to capture all those solutes that would be inside of these tubules. The fluid will be percolating through a long, winding cellular tube, and we'll be reabsorbing all those nutrients, all those solute molecules and ions, ultimately, and water from the tubules. So we got to have a chunkier cell to handle all those organelles to provide space for them. Before we go on, I'll also mention that exocrine glands that secrete their products into ducts that ultimately are released onto the surface of the body, such as sweat glands, mammary glands, the pancreas that produces digestive enzymes. They all are made up, the secretory component, the cells that secrete the product, are simple cuboidal epithelial cells. So they make up the kidney tubules, and they make up gland secretory units. Acini, they're called, secretory acini. Here's a simple columnar epithelium. You can see these tall, narrow, rectangular cells, and these cells are lining the intestines. They line the stomach and intestines, the gastrointestinal system. Gastro just means of the stomach. So these cells line that entire organ system, and they're cells that are specialized, like the simple cuboidal cells of the kidney tubules. They're specialized for transport. All of the nutrients in your diet, once digested, need to be absorbed from the lumen. This represents the space inside your intestine. It has to be absorbed across this epithelium into the tissue below, where there's blood capillaries and lymphatic capillaries ready to pick it up. And so this is an epithelium that needs, again, a lot of organelles, going to produce a lot of ATP, a lot of transport proteins on the surface. Indeed, we need so many transport proteins on the surface that these cells have what are called microvilli. They're like microscopic finger-like projections sticking up from the surface of the cell to increase the surface area so we can pack in more transporters that can capture various solutes and transport them across the membrane. Remember, the solutes have to be captured, like in the kidney tubules, one molecule at a time and transported across, and then another, and then another. And then water will follow by osmosis. So interestingly, I've read a shock and awe statistic that a brush border, it's called, on a simple columnar epithelial cell, whose microvilli increases the surface area 300 times over what it would be if this was just a blunt, surf-topped epithelial cell. Pretty impressive, so we can pack in a lot of transport proteins to make sure that we capture all the nutrients before it's too late, before defecation occurs. So in the next section, we'll pick up and look at another few epithelia as we go through. In fact, we'll finish up the epithelia and probably start on the connective tissue. So join me next time for part 2 of our survey of the various tissues that make up all the organs and organ systems of your body, chapter 4 of our anatomy and physiology text.